Hello everyone and welcome to Pocket Size Bio, Dr. Casco. In today's video, we're going to take a look at Campbell's Biology, Chapter 11, Cell Communication. And the subsections that we're going to take a look at are external signals are converted to responses within the cell. That's section 1. Section 2, signal reception. A signaling molecule binds to a receptor, causing it to change shape. Section 3, signal transduction. Cascades of molecular interactions transmit signals from receptors to relay molecules in the cell. Section 4 is cellular response. Cell signaling leads to regulation of transcription or cytoplasmic activities. And the last section, apoptosis, requires integration of multiple cell signaling pathways. Okay, so figure 1, as we see here, figure 11.1, has a good introduction on what is essentially the overarching theme of cell communication. We have this picture here where this impala is fleeing for its life, racing to escape the predatory cheetah nipping at its heels. The impala is breathing rapidly, its heart is pounding, and its legs pumping furiously. These physiological functions are all part of the impala's fight or flight response. We're going to take a look at that. Driven by hormones released from its adrenal glands at times of stress. What is the stress here? Sensing the cheetah and, of course, being followed by the, trying to get captured by the cheetah to be eaten, right? More specifically, More specifically, how does the cell signaling fuel the desperate flight of an impala? All starting with the senses, right? The cheetah, the impala picks up this perception, right, these brain signals from sensory receptors, sending this message to the brain. The brain will then signal a message to the adrenal glands to release epinephrine into the blood. This hormone will then produce a signaling cascade, starting from signal reception, where the epinephrine gets bound to a receptor, and that will relay a message through a series of molecules, ultimately to produce a cellular response. In this case, glycogen being broken down into glucose molecules, where glucose can now be used to as fuel, for the leg muscles during this time of stress. Evolution of signaling. So the book begins talking about how signaling can be seen in even simple organisms. One of these, of course, being bacteria, and research was done in the 1970s that even bacterial cells can signal to each other, right? So bacteria can signal and secrete small molecules within the environment, and these molecules can be detected by neighboring bacterial cells. That whole idea of being able to perceive a signal from one bacteria to another, to a population of bacteria, let's say inside a culture dish, right? It's called quorum sensing. And there's a little bit more to that, right? The concentration of signaling molecules, okay, so how strong that signaling molecule is in terms of concentration can enable bacteria to sense the local density of the population. So how many cells are in the close vicinity. This in itself is called quorum sensing. And as a result of that, that can lead to a coordination in their activities. One of the most common consequences of quorum sensing and this detection is producing a biofilm, right? Bacteria come together, they secrete this film of molecules of this film, okay? And then this biofilm can protect the bacteria that are inside it, okay? So, moreover, they're aggregations of bacteria, right, containing regions of specialized functions, and they can protect the cells inside from this kind of mucal membrane, if I can call it, not necessarily a membrane, right? The following page, I'm going to continue with biofilm. The following page contains this problem-solving exercise dealing with quorum sensing, where cells sense their own population, right? And at a certain density, they start to secrete a toxin. That's one of the properties that can be done only by the biofilm, right? Because you have a certain density. And then you have this problem-solving exercise. I'll let you guys work on that so you guys can see. Okay. So, this idea of biofilm can even help provide nutrition to the cells that it is kind of coating, right? Some examples of biofilms are slimy coatings that are on a fallen log, leaves lying on the forest path that have some kind of mucal residual, if I can say. We can even feel biofilm, when we wake up, unbrushed teeth, we can feel it in the surface of our teeth, right, are produced by biofilms. So, and like I mentioned before in that science practice, toxin secretions of infectious bacteria, right? This could have medical implications. Again, so you have this biofilm, you have this density of bacteria, and then since they have this density, they can secrete collectively a toxin, okay? And a problem with this also is that, especially, with bacteria that are resistant to antibiotics, and, they also will produce this biofilm and they'll secrete the toxin. So, because they have this density, right? But if science and if medicine can produce a drug that can prevent the biofilm from forming, that could be a possible form of treatment. If you disrupt the biofilm, then you can't create this densely packed population of bacteria to produce the toxin, right? So that could be another way to beat infectious bacteria, especially now that we're getting situations that several bacteria are resistant to antibiotics, right? And so you have this idea of signaling, right? Even simple organisms. You can also see it in yeast. This is another example of signaling, simple signaling, right? And in the case of yeast, we have this example of reproduction where Baker's yeast, Saccharomyces cerevisiae, they mate, of course, and to identify potential mates, they undergo chemical signaling. Again, another example of signaling here. So in yeast, there's two sexes. You have an A factor cell, okay? You have a yeast cell that produces an A factor. It's literally called A factor. So they're this mating type that they produce this particular factor. And then you have another type of yeast cell that is called the alpha mating type. The reason why they're called that is because they produce an alpha factor, so another X and Y type of factor there. The thing about this is that these factors, they're produced by these cell types, but they're only their receptor is on the other cell type. So if you see here, you have yeast A, it produces A factor. It has receptors there to receive a particular factor, protein, whatnot. But if you look here, the shape of A factor doesn't fit the surface of this particular cell. It fits the other mating type. Okay, so if you see, it's very circular here, and it's going to bind to the receptors of the other cell type of alpha yeast cells. And vice versa, alpha yeast cells produce the alpha factor, which can be received by the other cell type. So they're complementary to each other. So this enables the other to bind with the opposite or the other mating type, okay, and which ultimately will fuse together. Their cells will bind together and migrate together, and then you'll get a new, variant of genetics from this A and alpha cell. Their nuclei will fuse, and now you have a new cell type. Okay. So again, the cells grow toward each other. They undergo cellular changes. They have their nuclei fused together, and then ultimately their genes are going to be from both cells. Okay, so what happens here is that after that initial, binding of this factor to the receptor, okay, um, you have a series of events, and that's all what signaling is about, right? So when a signal is attached to a cell surface molecule, that surface molecule, the receptor, is changed, and it will begin a relay of signaling molecules, right, that will pass one message to another, to another, to another. And it's often called being transduced, okay? And ultimately will lead to a cellular response, some effect, okay? And there's three major steps in this whole kind of process. So you have signal reception, signal transduction, and then cellular response. All of that together ultimately is called a signal transduction pathway, okay? A signal transduction pathway. So we've seen it in yeast, a very, very simple way. Okay, mammalian cells also do this. Plant cells do this. Basically, living organisms have a way to pass down a signal, right? And this is coming along, 1 billion years ago, so quite a while back. So signaling systems, can be found in bacteria, in plants, in animals, in fungi, protists. Okay, so what's quite interesting, ancestral signaling, you can see this pathway, this behavior occurring at a molecular level ever since prokaryotic cells, right? Ancient prokaryotic cells. Of course, they have evolved into very, very complex pathways, and we're going to take a look at some. Okay? So still going along in some kind of properties of signaling methods. So you have local signaling and long distance signaling. Okay? And communication and communicating cells, they can be literally adjacent to each other, or they can be far apart. Okay? Multicellular organisms that release signals to target other cells, again, they can be tissues, right, or they're kind of knitted even together, or they can be far apart. So we're going to look at each of these. Cells may communicate by direct contact. So we see here, this is an example of direct contact that I'm talking about. Okay, so you have animal cells and you have plant cells. And if you remember from chapter 6 on the different structures of the cell, here you have gap junctions. Okay, there's different types of junctions between two cells. And you have these gap junctions that enable signaling molecules from one cell to go inside another cell and vice versa. You have some chemicals that are being released from the adjacent cell coming into another target cell. And the same thing happens with plant cells. So they can pass readily through the cytoplasm just because they're connected to each other. So there's flow within the cytoplasm. Okay? These signaling molecules, of course, are generally dissolved within the cytosol, which dissolves and diffuses from one cell to another. So you have direct contact. You can also have direct contact where you have some molecule in one animal cell, and that molecule can directly bind to another cell that has some type of receptor molecule, okay? And that can enable a response within that receptor molecule or a receptor cell, okay? So here you have an example where you have cell A, it has some types of surface molecule, it would directly contact cell B, having some receptor that will create a complex here that would enable an internal signaling pathway within the cell. Where do you see these types of things in embryonic development? An immune response, especially an immune response when we talk about the B and T cells, right? And the different type of immune cells. Stem cell populations also have this behavior. Okay? And then you have other cases where you have a signaling cell secretes molecules in the vicinity. So it's a local area. Okay? So signaling cells can release molecules that influence the local vicinity. Okay, not too far. Generally, they're growth factors. hormones or proteins, and in this case for animals, they have compounds that could stimulate nearby target cells to grow and multiply, to relay a signal. Okay, and this is the panel you see here. So this type of signaling, of local signaling, is commonly called paracrine signaling. Okay, where you can get numerous cells being affected by just one cell that is releasing some type of signal. Okay, so if you see here in panel A here, target cells, you have secretory vesicles that are releasing that signal. A special type of paracrine signaling is where you have, or synaptic signaling, let's be more specific here, occurring in nervous systems where you have one axon or one nerve cell that is relaying a neurotransmitter. Okay, it's in these specialized secretory vesicles, passing them down to the synapse area. The neurotransmitters are being released into this space here, into that synapse, where ultimately they're going to be received by some target cell and some target receptor. Okay, this is called synaptic signaling. Okay, so you have an electrical signal in a nerve cell. It will secrete a neurotransmitter, which will then carry a chemical signal to be received to a target cell. Okay. And again, and then you have plants. Plants is not as well as understood as animal cells, but nevertheless, there is studies that show this type of signaling mechanism. Okay, and then you have long distance signaling. Plants and animals use hormones for long distance signaling. Okay, in this case, you have what is called endocrine signals, right? Where specialized cells, they release hormones into the circulatory system. And then the hormones will travel through the circulatory systems, all throughout the circulatory system, okay, all throughout our blood, until it reaches a target cell somewhere else in the body, okay? And these target cells have to have special receptors to receive this hormone, okay? Plant hormones can also undergo this type of work. They have a vascular system, xylem and phloem, and they're able to move about these particular signals. Okay, and hormones and these regulators, these molecules, can vary widely in molecule size and shape and type, right? Just to give you an example here, right? So you have an endocrine cell that is releasing some hormone into the bloodstream, okay? And the bloodstream is carrying this hormone throughout the body to be, received by some receptor and some target cell. Okay, long distance. Though we're going to get back to this with adrenaline. Okay, let's continue on. An example of plants, you have the plant hormone ethylene, C2H4. Yes, the gas that this can promote fruit ripening, can regulate growth of the plant, and it's a hydrocarbon with only 6 atoms in total, right? So relatively simple, nevertheless, it can carry out a great effect. Okay, and then you have some other ones like insulin, which is a very large molecule, thousands of atoms that could pass, through, that can move about through the blood. And this particular hormone regulates blood sugar levels, okay? So now we know a little bit in terms of the types of signaling, getting into the stages of cell signaling, okay? For AP students, you don't necessarily need to know this particular stuff in italics. For undergrads, everything is good to go, right? Depending on your instructor there. So cell signaling was pretty much discovered by Earl Sutherland, okay? And he won a Nobel Prize on, this discovery, studying many molecules. Amongst these is epinephrine, right? So he and his colleagues, pioneered the understanding of cell signaling by investigating epinephrine and how epinephrine can break down glycogen into glucose to be used for skeletal muscle fuel, right? So we're going to take a look and be going down with his example as we're going down into the different types or stages of cell signaling. Ultimately, right, we know that epinephrine will undergo glycolysis, break down, glucose will be sourced from glycogen to undergo glycolysis, right, into the blood or be used as fuel in general. Okay, so epinephrine in itself is the mobilization of fuel reserves. Okay, so the way that it does that is by activating a cytosolic enzyme called glycogen phosphorylase. Okay, so in itself, if you were to get epinephrine and glycogen in vitro, okay, so in a tube, you put a certain concentration of epinephrine and glycogen in itself, nothing will happen. Epinephrine does not activate the phosphorylase directly. So that's one of the hints that Sutherland started to get to be able to activate glucose usage, right? It is not directly involved, but involves a series of intermediate steps. Okay, so we're going to take a look at these three stages now. So cell signaling involves three stages: reception, transduction, and response. Okay, so signals reception. What happens in this particular case? You have a chemical signal binds to a cellular protein, typically at a target cell surface or inside the cell. Signal transduction is the binding of a signal molecule that changes the receptor protein, initiating the process of signal transduction. Okay. Now, transduction may occur in a single step, or moreover, it occurs in a multiple relay of molecules, okay, a series of different molecules. Those that are involved are referred to as relay molecules, as you can see here. So you have relay molecule 1, 2, and 3, and then one goes after the other and after the other and after the other. Okay? And lastly, you get cellular response. So cellular response, the transduced signal triggers a specific cellular activity. Now, this could be many things. It could be gene activation. It could be the breakdown of glycogen into glucose. It could be cytoskeletal rearrangement. So it could be many, many things there. What's critical to understand for cell signaling is that these processes and very specific signaling pathways occur on the right cells at the right time and in proper coordination, so we can always try to be in homeostasis or quickly respond to something that is getting us out of homeostasis. So it's quite interesting how this works. Okay, so the following sections just talks about each of these stages more in detail. We're going to take a look now at signal reception. So this is section 11.2. A signaling molecule binds to a receptor, causing it to change shape. Okay. So the cell targeted by a particular chemical signal, okay, has a receptor protein on or in the target cell that recognizes the signal molecule. If you guys remember, similar to the ligand enzyme, that you get this substrate enzyme complex, you're having something similar where you have a binding site, you have a ligand coming in, but in this particular case, you have receptors. Okay. And these receptors usually are found on the cell surface, on the cell membrane, or over the outer cell membrane, the outer side of the cell membrane of the cell. And we're going to take a look at that, but it's not always the case. Okay, you can find them within the cell, intracellular signaling, and so forth. Okay, just to kind of review what a ligand is, right? A ligand is any signaling molecule that could bind to a larger molecule. Moreover, a receptor, okay? Receptor proteins. And then when these receptor proteins bind to a ligand, they themselves will change shape. Okay? And ligand binding can cause an aggregation of other receptor molecules to just come into the vicinity. But there's a lot of variation that occurs here. Okay? So as a result, that could lead to a series of molecular events inside the cell, signal transduction, which we're going to see later on. Okay? As I mentioned before, most signal receptors are plasma membrane proteins, transmembrane proteins. The ligands generally are, and this is quite important to know, they're relatively large, they're water soluble, so they need to be moving about within the cytosol. And they're generally too large to cross the plasma membrane in itself, hence the whole purpose of a receptor molecule. Okay, so let's look at the different types of receptors in the plasma membrane. So these transmembrane receptors, they are very crucial for cell signaling to maintain biological systems in animals, like I mentioned before. The largest family of cell surface receptors in humans is the G protein coupled receptors. Okay, there's around 800, more than 800 GPCRs, G protein coupled receptors. Again, the water soluble signaling molecules would bind to these receptor proteins. Okay, and then ultimately they will relay information from the extracellular environment into the cell via changing the shape of the receptors. Okay, so there's three major types of receptors. The G protein coupled receptors is one group, receptor tyrosine kinases, and ion channel receptors. Okay, so we're going to take a look at each of them in some detail there. Much research has been done in receptors because generally, since they relay a signal, whenever you have mutations on these receptors, you're most likely going to have some disorder or disease on the patient. So it's a very, very critical area of study because of their implications, right? It can lead to cancer, heart disease, respiratory diseases like asthma, and so forth. Okay? Cell surface receptors are approximately 30% of all human proteins, so that's quite a lot. It is a challenge to determine the structure of these receptors because they could be quite flexible, they change shape, so they're difficult to crystallize. Okay, so it has been an endeavor for scientists to kind of figure out the 3D structure. Okay, but nevertheless, scientists have been able to model many structures. This is an example of a G protein coupled receptor. Okay, and a mimic, a molecular mimic in this particular case. Okay. All right. So, taking a look at these coupled receptors here. So the first one we're going to look at is the G protein coupled receptor. Okay, and the reason why it's called a G protein coupled receptor is because it has a G protein on the cytoplasmic side of the cell membrane. Okay, if you take a look here, let's look at number 1. So you have the receptor here, and then you have a G protein that is in the inner membrane side of the cell, the inner side of the cell membrane. Okay, that's why it's called the G protein. So the reason for this receptor is because the G protein in itself binds to a GTP molecule or a GDP molecule. Let's take a look. Okay. So these proteins that bind to GTP, some of these, are involved with epinephrine and yeast mating factors and neurotransmitter, and there's so many types of receptors that they can vary in their binding sites, right? They could be similar in structure, even polypeptide sequence. they have 7 alpha helices that span the membrane, as you saw in the previous picture, but nevertheless, they have slight variations in their binding site. The G protein itself acts as a on and off switch to the receptor and to relay a signal there. Okay, and similarities, again, as I mentioned before, between many different types of proteins and even amongst the different organisms can suggest that this type of signaling mechanism evolved relatively early, with some variations happening as different organisms evolved over time. So it was one of the early signaling mechanisms. And of course, relaying with GTP and functioning with neurotransmission and so forth, it again does involve with human diseases, has some implication with human diseases. Okay, bacterial infections cause cholera and botulism. They interfere with G protein function. If you if you somehow inhibit or over promote, you can cause some disease or condition in the body. Okay, just taking a look, and I want to go one by one in detail with this particular situation of G protein coupling. So you have your setup here. You have the receptor, you have a G protein that is in an inactive form, okay, on the plasma membrane, and you have some enzyme here, right? So what happens is the following, right? You have attached but able to move along the cytoplasmic side of the membrane. A G protein functions as a molecular switch, like I mentioned before, that is either on or off, depending if it has a GDP or GTP molecule attached to it. So that's the whole reason why it's called a G protein. Okay. GTP, or guanosine triphosphate, similar to ATP there. Okay. GTP or guanosine triphosphate, when GDP is bound to the G protein, as shown above, the G protein is inactive. The receptor and G protein work together with another protein, usually an enzyme. Okay. Number 2, when the appropriate signaling molecule binds to the extracellular membrane, to the extracellular side of the receptor is activated and changes shape. Okay, its cytoplasmic side then binds an inactive G protein, causing a GTP to displace a GDP. So this will activate the G protein. Okay, so do you see how the GTP is replacing the GDP? Number 3, the activated G protein will then dissociate from the receptor, diffuses along the membrane, then binds to an enzyme, altering the enzyme shape and activity. Once activated, the enzyme can trigger the next step, leading to a cellular response. Okay? So binding of signaling molecules is reversible. So this is quite interesting too. Like other ligands, they bind and dissociate many times. So you have a ping-ponging of the G protein going left and right and left and right. Okay? The ligand concentration outside the cell determines how often a ligand is bound and initiates signaling. Okay? Number 4, the changes in the enzyme and the G protein are only temporary. So the G protein also acts as a GT pace, hydrolyzing its bound GTP to GDP and an inorganic phosphate. As a result, it can no longer activate the enzyme. The G protein will then leave the enzyme, which returns to its inactive state. So its play between the receptor and the enzyme will inactivate and activate the G protein. Okay, so the G protein is now available to be reused. Its GT pace function allows the pathway shut down rapidly when the signaling molecule is no longer present. Okay, so the next molecule that we're looking at is receptor tyrosine kinases, and this is a major class of plasma membrane receptors. And this type of receptor deals with enzymatic activity catalyzing transfers of phosphate groups. So that's their kind of main point there, to another protein from ATP. Okay, so the majority of these types of receptors is found on the cytoplasmic side, as you can see here. So let me just zoom in. All right, so you have a receptor tyrosine kinases, and then if you can see, the majority of the structure is on the cytoplasm side, and it functions as a tyrosine kinase. And what does that mean? Right, a kinase, basically you have a transferring of a phosphate group from ATP to tyrosine amino acid on a substrate protein. Okay, so you have these tyrosines here, and so you have these this part of this receptor that will come in and obtain phosphate groups. Okay, so this type of receptor tyrosine kinase is very effective when cells need to trigger several signal transduction pathways and cellular responses in bulk, if I can say. Okay, so this regulates and coordinates many aspects of cell growth and reproduction within the cell. Okay, and so one tyrosine kinase receptor dimer may activate 10 or more different intracellular proteins simultaneously, and we're going to take a look at that. Okay, so that's one big difference between the G protein coupled receptors. Okay, and we're going to see what that visually looks like. Abnormal RTKs, okay, receptor tyrosine kinases, they activate, abnormal ones activate despite ligands to come in and activate them, right? So they are greatly associated with cancer. Excessive levels of RTKs called HER2 on breast cancer cells, it correlates with poor prognosis with patients. Okay, so the more you have of this particular kinase, HER2, the more intense the cancer, for example. Okay, so using molecular biological techniques, research have developed a protein called Herceptin that binds to HER2 on cells and inhibits their growth, so it reduces tumor development. So very, very important for medical treatments, right? Herceptin has improved patient survival rates by more than 1/3. So there's still a lot of research. So this is a good topic if you want to get into medical sciences, right? Studying signaling pathways. I myself did my PhD on signaling pathways with tissue regeneration, particularly with TGF beta 1 and BMP2. Okay. On cell surface receptors and other signaling proteins is development of successful treatments. Okay, so just to take a look at what exactly is happening in this particular receptor. So for tyrosine kinases, for RTKs, you have these protein structures. Okay. What happens in this particular case is that these receptors, they are going to get activated with a signaling molecule, but they actually, in order for them to activate together, you need two of them. Each receptor by itself is called a monomer, okay? But when two of these monomers get a signaling molecule and bind, they actually bind together. So these two independent ones will bind together to create a dimer, okay? So before the signaling molecule binds, the receptor exists as individual units. They're referred to as monomers, okay? And they each have an extracellular binding site, okay? And they have that intracellular structure here, which is that tyrosine's location. And then when you have that binding of a signal molecule, usually it's a growth factor, okay? It causes two receptor monomers to associate closely to each other, and they form what is called a dimer. Okay, and that process of creating a dimer is called dimerization. More clusters could form depending on the type of receptor. Okay, so the details of monomer, we're gonna take a look at that and dimerization later on for signaling pathways and for regulation of signals. Okay, and then what happens is that this dimerization will activate the tyrosine kinase area, okay, of each monomer, and then each tyrosine can add a phosphate from an ATP molecule to itself, okay? Which is, that tail part. So in this case, you have 6 tyrosines, and ATP will come in, phosphorylate the tyrosines, and now you have an activated receptor. When the activated receptor has the phosphates, it can recognize other relay proteins, okay, that can later induce some cellular response. Okay, so you have relay proteins that are in inactive form, okay, and then as they bind to a tyrosine that is phosphorylated, okay, they activate and induce a cellular response again. Okay, so now that the receptor is fully activated, it is recognized by specific relay proteins inside the cell. Each such protein binds to a specific phosphorylated tyrosine, so it gets very specific, undergoing a resulting structural change that activates the bound relay protein, and then that protein will trigger a signal transduction pathway. Okay, so these are RTKs, receptor tyrosine kinases, and we have a third one, which is ion channel receptors. Okay, so these are ligand-gated ion channels. They're a type of membrane protein, as you can see here, and a membrane receptor, and it can act as a gate, meaning that this channel can open and close, okay, via the changing shape of the receptor. So a signal molecule binds as a ligand to the receptor, and it opens and closes depending on the particular type and the particular type of receptor. An example of this are sodium, potassium ion channels. Okay, so binding by a ligand at the extracellular site. So remember, you're always getting signaling molecules from the outside. You could get the inverse though, so don't get me wrong in that. But usually it's from the outside coming to the inside. Okay, protein changes shape, opens the channel. Okay, very specific examples are for neurotransmission, where you have in the nervous system neurotransmitter molecules released at the synapse. So if you remember what I was talking about, the synaptic signaling there, right? Between two neurons bind as ligands to ion channels on the receiving cell, causing the channels to open. The channels open, ions can flow in and trigger a difference in active potential, right? In action potential. And then you have that electrical signal that will propagate down the length of the receiving cell, and you would close and open ions. So you have an influx coming in and going out and so forth, okay? Depending on the concentrations there. Okay, so just to give you a sense here on the ion channel receptors, okay? Let me just move this a little bit here. So here's a ligand-gated ion channel receptor in which the channel remains closed until a ligand binds to the receptor. Signaling molecule comes in, okay? When it binds, specific ions can now flow in because the protein has changed shape. In this case, it has opened. So specific ions can flow through the channel, rapidly change the concentration of that particular ion. Okay? Activate a signal response in some cases. When the ligand dissociates from the receptor, the channel closes and no more ions can come inside. Again, so this is ion channel receptors. All right, and then you can get intracellular receptors, right? So we've been talking about receptors that are on the membrane of the cell in the extracellular side, but you can also have receptors that are inside the cell, okay? They can be found in the cytoplasm or nucleus of the target cells. Okay, so chemicals pass, again, all the time through the plasma membrane if they are hydrophobic enough, if their size is adequate enough, right? They can cross through the phospholipid into the plasma membrane. Okay, some examples could be steroids, okay, hormones, steroid hormones or hormones coming from the thyroid gland of animals, okay? Even some signaling molecules could be nitric oxide, right? A gas whose the small size of this gas allows it to pass between membrane phospholipids, which can actually have some implications with signaling. Okay, so an example of this type of intracellular receptors is aldosterone. Okay, so it is secreted from the adrenal gland. Okay, so it travels through the blood. It's secreted in the adrenal gland, goes into the bloodstream, passes through the whole bloodstream. Okay, all the cells in the body. But if you don't have the receptor, nothing's going to happen, right? So only kidney cells respond because they have the adequate receptor protein. Okay, and whenever they bind to the aldosterone, it binds to this protein. Binding activates the receptor protein to enter the nucleus. Okay, so it acts as a receptor and a transducer where it's coming into the complex there. And then that could turn on specific genes within the nucleus. Okay, in this particular case, genes that are related to water and sodium flow. Okay, and then that ultimately affects blood volume. So something very interesting as we talk about gene regulation in the later chapters, these complexes, right, these activated proteins within this signaling pathways, some of them can act as transcription factors, okay, TF. So we're going to see this a lot more in detail in the later chapters. And these transcription factors control which genes are turned on or turned off. So it would regulate the process of transcription, ultimately creating mRNA, right, or messenger RNA. Very, very interesting subject there we're gonna get into there. Okay, so this is an example of what I was talking about. You have this hormone, aldosterone, that it diffuses, where you can just pass through the membrane. This membrane comes in and it could bind to a particular receptor protein that's in the cytoplasm, makes this complex, and that itself will kind of shuttle it. Making this complex will shuttle it into the nucleus. Once it's in the nucleus, it will bind to specific genes, so it can act as transcription factors to initiate transcription, therefore creating mRNA. And then that mRNA will, of course, later go on to be further processed, spliced, exit out of the nucleus for translation to occur to produce a particular protein. Okay. So this is 11.2, and in the next video we're going to go into signal transduction. Okay, the cascade of molecular interactions transmits signals from receptors to relay molecules in the cell. Very, very interesting concepts here and processes that we're